Are you seeing my screen, Maha?
Yes, I'm able to
Perfect.
see your screen and everything, yes.
Perfect.
So the attendees have started to join. We have 4 attendees so far.
Perfect. Hello, Miss Jima. Miss Jima is here.
Yes. So we'll just give a couple of minutes to let the attendees join the session, and then we'll start with our presentation.
Sure. So good afternoon, everyone. As Miss Maha said, and thank you, Maha, for being here. We'll be waiting for 2 more minutes until the other attendees will join us. Thank you.
We are still receiving attendees, so just 2 more minutes. At 4:05 exactly, we'll start the session.
Sure. Sure, Maha. Thank you.
You're welcome. I think, Dr. Tanya, we can start now.
Okay, thank you, Maha. So good afternoon, ladies and gentlemen. My name is Tanya Tahtouh, and welcome to this presentation. Today, we will have a peek inside our brand new labs. So let's get started. So this is the structure of the presentation. At the end, we will be discussing important information concerning the students' learnings and career opportunities. And please take notes as we go, because at the end, we will have also a Q&A session. Sorry for this. So in this presentation, we will be using an interactive website. The link is www.menti.com. It's like Kahoot. I'm sure you are used to use Kahoot. And please use the code 5310910 to enter this activity. You can go to any web browser on your phone, on your computer. Just write menti.com, add the code, and be ready to start the activity with us. So hello again. My name is Tanya Tahtouh. I did a PhD in cell biology, and the courses I teach are biochemistry, biotechniques, human biology, clinical biochemistry, and enzymology. So the programs in the College of Health Sciences include the Bachelor of Science in Biomedical Sciences, Laboratory Medicine, Bachelor of Science in Molecular and Medical Genetics, and Bachelor of Science in Human Nutrition and Dietetics. So these three programs have begun under the umbrella of the College of Health Sciences in 2019. And since that time, we have been refining and adding to this curricula. In support to these programs, ADU has built 4 labs. Dr. Wassim Almawi, the professor and the dean of the College of Health Sciences, and I have been designing and setting up these labs. Ms. Dima Al-Halabi has added an integral role in establishing, has played an integral role in establishing the nutrition lab. So here are the 4 labs: general lab, molecular biology lab, nutrition lab, and special chemistry and histology lab. These labs have been designed to help students experiencing new concepts from quantifying DNA, protein, to body composition analysis. So this is the layout and the design of the general lab. In this lab, students will get a taste of real-life research. The general lab specifically supports students from biomedical sciences, lab medicine, and molecular and medical genetics programs. The capacity is for 20 students. This is the molecular lab. In the molecular lab, students will experience and do some research in DNA and RNA analysis. This lab supports students from biomedical sciences, lab medicine, and molecular and medical genetics programs. Its capacity is for 15 students. This is the nutrition lab. In this lab, students will learn how to analyze body composition. They will learn about proper food preparation, food safety, servings, and correct association of food in menus. This lab will support students from human nutrition and dietetics programs. Its capacity is for 15 students. This is the special chemistry and histology lab. In this lab, students will perform a variety of routine immunoassays, and we will watch a short video explaining what is an immunoassay, some HPLC and other manual tests. This lab supports students from biomedical sciences, lab medicine, and molecular and medical genetics programs. It can also support students from the nutrition and dietetics program. Its capacity is for 15 students. So, are you curious to know which equipment, which machines we have in our brand new labs? So this piece, the Luminex 200, is in our labs, and it's used to perform up to 100 immunoassays at the same moment. So what is an immunoassay? This video will explain more about immunoassays.
Immunoassay. The purpose of this animation is to explain how a prototype example of this assay works. The enzyme immunoassay can be done in a multi-well microtiter plate or on any other solid adherent surface. I will use the microtiter plate in this example, so let's take a closer look at one of the wells of this plate to see what happens during the performance of the assay. The plate is prepared to perform a particular assay by coating the wells with antibodies that bind to the antigen of interest. Then the wells are filled with the clinical sample, which could be a sample of serum, respiratory secretion, cerebrospinal fluid, urine, or some other body fluid. If the antigen is present in the sample, it will bind to the fixed antibodies. In this example, the green shape represents the antigen of interest, and the other shapes represent other molecules in the sample. However, note that only the specific, or green antigen, and none of the irrelevant molecules bind to the antibody-coated wells. This accounts for the specificity of the test. The wells are then washed out to remove any of the unattached molecules, leaving the antigen of interest stuck to the wells. Now, a second antibody, directed against another epitope on the target antigen, is added. These antibodies are conjugated covalently to an enzyme, indicated by the yellow circle at the Fc portion of the second antibody. They bind to the antigen that's fixed in the well, and this provides a second level of specificity for the assay. The wells are washed again to remove any unbound antibodies, and in the final step, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate and the captured enzyme generates visible color in the solution. At the macroscopic level, the development of color indicates those samples that have second antibody bound to the target antigen in the wells. Thus, the wells that change color are the ones that contain the antigen of interest. In other words, the positive tests.
Okay, so please go to Mentimeter. As I told you, please try to access it from your phone or your computer. Write only www.menti.com and use the code 5310910. And try to answer the first question. How excited are you to know more about our labs? So I assume that you are excited. to know more about our labs so we can continue. So now we will proceed with my presentation. This is a power lab, and it's a simple and flexible way to collect physiological data. So do you want to know more about how does it work? Let's watch again another short video explaining how a power lab does work. Can you hear the audio coming from my computer?
No, doctor, not yet.
Okay. But before, in the previous video, Maha.
Yes, doctor, we could hear it clearly.
Excellent. Thank you, Maha.
Hi, I'm Dr. Ivan Matthews. I'm here at Montclair State University in the Exercise Science Lab to show you how to use a PowerLab hardware setup with LabChart software and the ADI sort of finger pulse pressure device that will actually come with this hardware. So it's fairly simple to use, so let's go ahead and get right to that. So if you just take this and you plug, so this is a DIN pin connector, and you plug it into whatever channel you want. So I'm going to plug it into channel 1 here, because that's where I'm expecting to see it in the PowerLab software. And it'll get a little green light that lights up on the plug when you plug it in. All right, so this little guy, which is the actual finger pressure or pulse pressure transducer, has a little sort of tacky, soft side to it, and then the other side is covered in Velcro. So get that tacky, soft side on whichever finger you're going to try to measure the pulse from. And what you're going to want to do is you're actually going to want to wrap this around fairly tight and then get the Velcro to stick. So nice tight wrap, get it on there almost as tight as you can. Obviously, you don't want to break or damage the device or hurt the individual who's wearing it, but the tighter you get it on, the cleaner the signal is going to look. You can also do this by, say, if you put a blood pressure cuff on, sort of stuff it up into that blood pressure cuff and then inflate the cuff to a level high enough where there's nice tight pressure on the arm. That actually will do probably a better job than putting it on a finger, but you just want to make sure that you're not occluding any blood flow when you do that. All right, so now that we have everything hooked up and I'm actually connected with the device on my finger, I have the PowerLab turned on in the back, which is a little green switch in the back. Now we're going to open up the LabChart 8 software. So now it's going to be scanning, looking for the devices. So it's specifically looking to make sure that the PowerLab is connected and turned on. And you can see here it's got the green check, so it is turned on and it is connected properly. These other ones we're not going to be using, so let's just ignore that for now and go ahead and click OK. All right, so here we have our different settings files, which you could build this into a settings file, but at the moment I don't have this built in. So let's just go over here and click on New, which is going to open a new document with no previous settings programmed into it. So here's our document. I have the device plugged into channel 1, so I'm going to make channel 1 really big and just sort of scrunch the rest of those channels that we're not using at the moment. All right, so all we have to do is hit the start button, and you're going to see that data is going to start recording. So there it is. There is my pulse pressure going into my finger. You can make this a little bigger with these plus signs, or this plus sign here, or you can make it smaller, obviously, with a negative sign. And you can see the pulse coming with each heartbeat. So you want to make sure that the person who's doing this is staying nice and still and relaxed. So it's probably best if they're laying down doing this so that they're not moving or supporting their own body weight by sitting up like I am. Because if the person moves in any way, I'm going to move my hand now, you're going to see all kinds of noise to that signal. All right, so you want to make sure they're nice and still like I'm now doing again, and you're going to get a reasonably clear signal. So this can be used for calculating heart rate, doing heart rate variability. Some people might actually try to use this to do some sort of systolic blood pressure assessment, but it's probably not great for that. But you can see that the peaks of each one of these pulses is going up and down depending on my breathing cycle, which is because it is actually linked to your blood pressure. So I hope that was helpful. Again, very quick, very easy to use.
So I hope that was helpful and you understood now what is the main applications for this Power Lab. This is the Neon Transfection system. It enables fast and efficient delivery of nucleic acids. I'm sure you heard about nucleic acids, like DNA, into all mammalian cell types. Imagine human cells, like breast cancer cells, hippocampal cells, including primary stem cells and difficult to transfect cells. This particular equipment or machine is the Transplant turbo transfer system. Imagine that you can transfer protein to membrane, which you can see here on this photo. Can you see the membrane with pink and blue colors? In as fast as 3 minutes, you will transfer your protein from a gel to this membrane in 3 minutes. This particular machine is the sequencer. How does it work? So imagine that you can sequence your DNA to find out what illnesses you might be prone to. Then you could custom tailor treatments and cure to yourself. So have a look at this slide. Please take 1 minute because after this slide, I will ask you to go to Mentimeter. I prepared a question for you there related to this slide. So focus a little bit, take 1 minute and try to find out some answers mentioning in this slide. We are still talking about the sequencer. So we'll progress with the Mentimeter question that I prepared for you. So what is Sanger sequencing? A way to read foreign alphabets, a method for sequencing Mr. Sanger's DNA, a method for determining the nucleotide sequence of DNA. You can start voting. Voting is closed. Let me repeat it, please. Sorry for this. Yeah, please go ahead and try to participate. What is Sanger sequencing?
Doctor, the voting is still closed.
Thank you, Maha. Voting is closed. Let's go back. Sorry for this. Wait, please. Ma, I will stop sharing my screen for a moment, please.
Sure.
Hello, everyone. I would like to know why the voting is closed. Settings. Sorry, ladies and gentlemen, give me 1 more minute to try to figure out the problem. Only for this one, it's closed. I will repeat it, maybe duplicate it. Now again. It's okay, maybe we can move on to the next question. But for this one, if you can write in the chat box, maybe the answer, please. Yeah, voting is closed for the second one. I don't know why. Only for the second slide. It's okay. So I assume that you know the answer. Let's see if you answered in the chat box. Sorry for this. Somehow I will be sharing again my screen.
Yes, doctor. So we can answer, the attendees can answer in the chat.
Yes. So the question is, what is Sanger sequencing, ladies and gentlemen? Did I have?
Yeah, we got two answers, and both of them are saying it's option A.
In the chat box, okay. Thank you. Okay, A. What was the A? A way to read foreign alphabets. Think about it. Is it something related to alphabets or to DNA? Thank you very much for participating. Okay, so kindly go to Mentimeter. Okay, so let me give you the answer. What is Sanger sequencing? It's a method for determining the nucleotide sequence of DNA. So you will know what is the sequence of your DNA, of your genome. Everywhere the vote is closed. It's okay. We'll see what we can do for this. You cannot subscribe to the Mentimeter. Only allows one free vote. Yeah. Thank you, Dr. Alia. Okay, let us continue now. This particular machine is, you can see, I think, my screen. So this particular machine is the Quantstudio 6-Flex real-time PCR. So with this machine, you can do a lot of RT-PCR, real-time PCR, genotyping and genomic profiling, digital PCR. You know that one of the coronavirus tests is based on RT-PCR. So this animation video is about the standard coronavirus test, which is the RT-PCR. Are you ready? Are you curious to know what is this about? What is an RT-PCR? Mahab, please, if you can confirm if you can hear very well the audio coming from my computer.
Severe acute respiratory syndrome coronavirus 2.
Is it working?
Yes, all good.
When a
Thank you very
person
much,
is
Mahab.
infected, the most common symptoms include fever, cough, and shortness of breath. The samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx, then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature. Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained. After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any remaining impurities from the membrane, leaving only the RNA bound to the silica gel. Once the sample is washed, the column is placed in a clean microcentrifuge tube, and an elution buffer is added. Then, a centrifugation is carried out, forcing the elution buffer through the membrane.
So for now, it's only the preparation of the sample. Okay, how we will extract the RNA, and then we will see what will happen.
The elution buffer removes the viral RNA from the spin column. And a purified RNA, which is free of protein, inhibitors, and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a pre-mixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used.
So let me comment on this. If you don't have the coronavirus in your sample, the RNA of the coronavirus, you will never read it. So this step will never happen. Okay? You will never add any nucleotide facing to the RNA virus sequence. Okay?
Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius. and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal,
for just $67, you can make as many videos as you want.
So I hope you enjoyed this video and you understood 80% of what is an RT-PCR and how we are conducting a coronavirus RT-PCR. So I don't know, Menti isn't working, but let me see if I can do anything for this, or at least I will address the question that I prepared. Oh, maybe it's working now. It's working. What does PCR stand for? Please, can you go to Menti and try to answer this question? Maybe it's still not working. Ah, excellent.
It is working, doctor.
Excellent, excellent. Thank you, ladies and gentlemen. Thank you, Maha. Thank you very much. Excellent, thank you. After this short video, I'm sure that you know what is a PCR. Thank you. Let's see what I prepared other than this question to you. Now we will move on to the last few machines that I want to share with you. These are some of our machines, okay? For example, this piece is a PCR system. You can run on this equipment 3 PCR experiments at once, okay? This is a camera, an eye bright imaging system. So what you can do with this camera is you can capture data from protein gels, nucleic acid gels, and have a look at the photos that you can get from this equipment, from this machine. So these are the photos of the proteins. Here you have, for example, many proteins. Here is a specific protein. This equipment is for the nutrition lab. It's the Embody 770, and it provides the most extensive analysis of the body composition. Now, are you ready to visit or have an idea about our labs? So These images are from our brand new labs. Please pay attention, and because after each lab, I will ask you to name the lab on Mentimeter. So have a look, try to find out which machines are in this lab to answer the question that I prepared for you on Mentimeter. So this is a sequencer. This is the machine for the RT-PCR. So go to Mentimeter, please, and try to name this lab. You just saw some photos from our brand new lab. Which lab is this one? Excellent. It's the molecular biology lab. Yeah. Perfect. Maybe I will give 10 more seconds to the other. Thank you. So now have a look at these photos and try to find out in which lab we are. I'll put them back. Okay, please try to go to Menti. You have a question that is ready for you there. I think you know the answer. Focus very well, ladies and gentlemen, and try to find out in which lab we were. We were visiting a virtual visit. Which lab? Perfect. It was the general lab in which we can do protein analysis, quantification of protein. You saw the transplant turbo equipment, a lot of vortex machines, the big freezers. Okay. Now have a look at these photos, please. I think it will be easy for you to answer the question on Mentimeter that I prepared for you. Name this lab, please. Perfect. We were in the nutrition lab. Excellent. Thank you. Thank you for your participation. Perfect. So now, are you ready to answer some questions regarding some career opportunities? I prepared some questions on Mentimeter too. So please try to answer the first question. You have two questions related to this topic. What kind of experiments and data would you like to collect in these labs? Do you want me to help you? Like, What do you like to experience in these labs? About cancer, excellent. Thank you. Nutrition, yes. What about genotyping, sequencing DNA, find some diseases? DNA. What about purifying some proteins? Purifying DNA, RNA? Test your blood pressure, maybe. Okay. I'll put another question. Thank you. Thank you very much for your participation. What careers would use devices like the one that I showed you before? Research. Perfect. Thank you very much. Forensic studies. Yes, excellent. What else? Inventions, experiments. Perfect. Thank you. Laboratory technicians. What about dietitian? Blood sampling. Excellent, thank you. Inventions. Collection. Nutritionist. Excellent, thank you. So all these answers are relevant. Thank you very much. And I hope that my presentation has helped give you an idea of the pathways that ADU College of Health Sciences can open for you. Pharmacists, analysts. Yes. Thank you very much. There are some other career opportunities, or maybe you mentioned most of them. Academic researcher, biotechnologist, biomedical scientist, forensic scientist, clinical research associate, dietitian careers. So thank you very much, ladies and gentlemen. And it's time for the Q&A session, portion of this session, please. I'll stop sharing my screen. Maha, do we have any questions?
No, doctor. Just give me a second. Let me review.
Thank you, Dr. Ali. Thank you very much for attending this presentation. Do you have any questions? So far, I can't see. I can see. Yeah, tell me, please.
We have Dr. Wasim raising his hand. So would you like to allow him to talk?
Sure, please. Please, Dr. Wasim, thank you very much for attending this presentation.
Dr. Wasim, you just need to unmute yourself.
Okay, so now I'm unmuted.
Yes, yes, doctor.
Okay, thank you very much, Dr. Tanya, for this really great presentation.
My pleasure.
I could not have done it better myself.
Thank you, Dr. Wasim.
And it was the hard work that the team put together. I know you don't like to take credit, but most of the credit goes to you. My question to you is, what will be, if it were up to you, what would you like to add to the existing facilities that we have? It could be either a new lab, or it could be a new system, or new equipment, or new lines of research.
Thank you very much, Dr. Wasim, for your support. I couldn't done this without you, first of all. And I already maybe mentioned it long time ago. I want really to add to our labs a cell culture room, a facility in which we can culture some cells and add some medicines and see the effect of this medicine on, for example, the breast cancer cells or hippocampal human cells, and find out the toxicity, first of all, of these drugs and the mechanism that these drugs can do in the cells, in human cells specifically. For sure, we'll not have, I think, animals, mice in our labs, but why not? Because all the experiments will start on animals, then on humans. And first, we can really in parallel work on cells, human cells, like the preclinical studies of every medicine, of every drug.
Well, I think everybody will be happy to know that your wish will come true, inshallah, over the next few months. We will be having our own dedicated tissue culture facility.
Thank you. I'm very happy to hear this. Thank you very much.
Thanks. And I'm done with my questions.
Thank you, Dr. Wasim. Ms. Maha, I can read some questions. Like, do students of nutrition cook in labs as well? For now, we don't have a cook lab, but we are thinking of really having it. I'm sure that Dr. Wasim and Ms. Dima, if she's attending this presentation.
Yes, Ms. Dima would like to say something, so I will allow her to speak.
Yes, please. Thank you. Thank you, Ms. Maha.
Yes, hello. Can everyone hear me?
Yes, yes, we can hear you, Ms. Dima.
For those who are interested in knowing more about the culinary labs, we are currently in the process of planning our new labs related to culinary, because we do take a culinary course, which is quantity foods, which is learning how to cook on a large scale, like they do in food service facilities such as hospitals, medical care centers, restaurants. So we are in the process of building those labs under the leadership of Dr. Wasim. The food science labs, they will take place in the same. So we also have food processing and culinary together. So this is a very near future project.
Thank you, Ms. Dima. Thank you very much for your answer.
Yes.
And there is one more question. If students need to test something like analyzing PCR results, can they collect volunteers from other colleges? So far, we're not really doing any real experiments in our labs for the coming, I think, few weeks because of coronavirus situation. You know this. So why not in the few coming maybe months or weeks? I'm sure Dr. Wasim will be happy. I will be happy to have students in our labs and to collect samples from our colleagues too. But so far, we're not doing any real experiments in our labs for the coming only few weeks until a further notice regarding coronavirus situation. So that's all I think for the questions. Please don't hesitate to add your questions or text me or send me any private message by email or.
Dr. Tanya, we have an attendee who would like to ask us another question. Should I allow her to speak?
Sure, please go ahead, Maha. Thank you.
Yeah. Thank you.
I may also add to what you said, Dr. Tanya.
Hello, Dr. Tanya.
The only drawback is we are still waiting for the consumable items to finish the labs. Once we have all the consumables in place, which inshallah should not take more than a month and a half, we will be ready to have a soft start of the labs. And eventually we will be doing real experiments using real samples, and starting with me and other students in our college and neighboring colleges, and also in the community and outreach. So these are going to be functional labs, and it's going to be for everyone to benefit from their capabilities.
Thank you, Dr. Wassim. Yes, someone asked or tried to ask a question. Sorry, I couldn't hear you before. Can you repeat your question?
Assalamu alaikum, Dr. Tanya. It was a very fruitful and knowledgeful session. I'm not a biological student, but I just want to know something about something new. That are you doing experiments nowadays, or are these experiments useful? Can robots can do that?
Robots. Okay, I'm doing with my colleagues in collaboration with other institutes outside of the UAE. I'm still really analyzing and still performing some analysis on experiment that I did. Right now, as I told you, we cannot really perform any real experiment in our labs only for the coming few weeks. Can we replace the human by a robot? This is your question. It can be when we are doing a lot of samples. Like I used to screen per day maybe 1,000 samples using a robot. Yes, but first you should try to do this experiment by yourself and then teach the robot and enter all the data, like make some program to the robot explaining all the steps that you did and that you tried manually. And then you have to adjust the place of every solution and put the number of tips, the number of rows should be correct. If not, you will break the robot, sorry. So it's very, very delicate, but yes, for some of the experiments, yes, because it will be faster, it will be more accurate, but you will still use the human skills for sure.
Yeah, robots cannot replace humans.
Yeah, no, never.
No,
you have to teach the
so
robot.
much.
You have to enter the program to the robot.
Yeah, exactly. Oh, thank you so much, ma'am.
My pleasure. Thank you. Thank you for attending this presentation.
Oh, thank you so much. Thank you.
Any other question before we end this session?
I think, Doctor, we're done. We have answered all the questions. If there are any other questions, please feel free to ask.
Ms. Maha, thank you for your support. Professor Wassim, thank you very much for being here and for your support, as well as Ms. Dima. Thank you very much.
Thank you very much.
You increased my knowledge, ma'am. Thank you so much.
My pleasure. My pleasure. Anytime. Okay, I think, Ms. Maha, we can end this session.
Yes, thank you so much, everyone. Hope to see you soon.
Have a great afternoon. Thank you. Bye.